Dear colleagues, ladies and gentlemen, welcome to this EASL webcast on the virological aspects of COVID-19. My name is Darius Moradpour from Lausanne University Hospital and the University of Lausanne in Switzerland. I'm with two world-renowned experts, Professor Jan Felix Drexler from the Institute of Virology of the Charité in Berlin, Germany, and Professor Volker Thiel from the Institute of Virology and Immunology from VetSwiss faculty of the University of Bern, Switzerland. We are in the midst of the coronavirus disease 2019, the COVID-19 epidemic. Felix, my first question goes to you. Where did the SARS coronavirus 2, SARS-CoV-2, the cause of COVID-19, originate from, and why does this coronavirus spread so rapidly and widely?
We have a huge diversity of SARS-related coronaviruses in bats, and more specifically so in bats of a certain genus, which is the genus Rhinolophus. So these Rhinolophid bats host an impressive diversity of SARS coronavirus strains that are not necessarily, for all we know, the direct ancestors either of the SARS coronavirus that emerged in 2003, 2004 in China, or the virus that is causing the current pandemic. But the sheer diversity of those genomes and their phylogenetic clustering strongly suggest, and I would dare to say provide definite proof of an evolutionary origin of SARS coronaviruses in those bats. And the big question is it likely that humans acquired the virus directly from a bat? Now, we know that bats are consumed as bushmeat in large portions of the tropical regions of Africa and Asia. And bats are also used in traditional Chinese medicine. And we know that both SARS and SARS-CoV-2 emerged in China. But the direct contact between humans and bats is rare. And the big question is, do we have intermediate hosts that could not only serve as a source of human infection, maybe in a wildlife market setting, and potentially, and very hypothetically speaking, even adapt those viruses for human infection? And I think we have a slide here showing that the whole scenario is not unique for SARS coronaviruses. Indeed, you have several coronaviruses infecting humans, and there's 4 viruses that are circulating all mainly in the winter season, so they're always infecting people. And these have you see them on the top part of the figure that we have prepared for this. And then we have the zoonotic coronaviruses, so SARS, SARS-2, and MERS coronavirus. And for many of them, you have the same picture. You have an evolution in an animal reservoir, mainly bats, not always though. And then you have intermediate hosts. And these hosts, for example, could be livestock. So there's maybe a virus, one of the viruses infecting us was maybe once a virus infecting cattle. And then it became a human coronavirus, and it's now passing between humans. Or in the case of MERS, you have a very clear picture in that human infections are almost exclusively acquired from camelids. So really, this is the big question. And what could be the intermediate host in SARS coronavirus 2, if there is one, is I would say not entirely clear yet. And we've also prepared a slide for this, which shows the pangolin theory, which is the best theory we have by now. And this is a simplified slide from a very nice paper done by our colleagues. And it shows you a simplified phylogenetic tree where the human virus is put in relationship with a virus from a bat and a pangolin virus. Pangolins are the suspected source of SARS-CoV-2. But as you can see on those 3 simplified trees, the picture is not quite clear, because the virus from the pangolin is somewhat closely related to the human virus only in the receptor binding domain, which is the middle part of the tree. Whereas in other critical genomic properties, the human virus, the virus infecting humans causing the pandemic, and the pangolin viruses, I should say, because there's now 2 divergent lineages, are not closely related at all. So there's no proof that the virus that is currently infecting us is of direct pangolin origin, and we still don't know that. But what we do know is that this is definitely a zoonotic disease, so we can see the origin in wildlife, and this also helps us to maybe refute hypotheses on an engineered origin of that virus. So this doesn't look at all, even beyond looking at virological properties of the genome of the virus currently infecting us. If you just look at the evolutionary history, you have a very clear picture of a zoonotic virus that is not driven by a crazy virologist like Volker in the lab.
Well, thank you very much, Felix. With this, I would like to hand over to Volker and ask you, Volker, what are the key virological features of SARS-CoV-2? And what kind of model systems do we have to study this virus, to learn more about its life cycle, pathogenesis, and eventually also to develop new antiviral approaches?
Yeah, Darius, thanks a lot. We had seen very early on from this outbreak the sequence of this new virus. It was published very soon, and we saw immediately that this is a virus that is really, really closely related to the SARS coronavirus that we have seen in 2002 and 2003. And as Felix already pointed out, this new virus is not engineered, because if you look at the sequence, about every 10th or 20th nucleotide is different, so nobody could ever construct this. So that's just a comment to Felix's point here. Of course, since they are so closely related, we can use a lot of systems, experimental systems, and also knowledge that has been gained already on the work on SARS coronavirus 1. And maybe just to point out a key difference between these two viruses, although they use the same receptor, as we can see here on this slide, which is ACE2, they are phenotypically, when we look at disease progression, but also even more pronounced on transmission, they are quite different. So they use the same receptor, but one virus is more causing, in more cases, severe disease, and the new coronavirus is not causing so many severe disease cases, but it's transmitting really, really well in the human population. So despite these viruses are so similar, and we see a couple of domains that are indicated here, the replicase complex ORF1A, ORF1B, they are very, very well conserved. We already talked a little bit about the spike protein that is very similar, but in some positions very different, or has at least phenotypical differences. And we see that all the accessory genes that are here shown in red, with ORF3b, ORF6, for example, ORF8, but also the others, ORF7, ORF3, they are also there in the new virus, but some of them show a little bit different sequence, like ORF8, for instance. So there are overall quite some similarities. It's really, these two viruses are really closely related, but there are also differences. And since they use the same receptor, we can build upon cell culture systems that we have developed for SARS. Also, primary epithelial cultures can be used because it's a respiratory virus. And essentially also antibodies, some antibodies that have been used in the past to study SARS coronavirus 1, they cross react with SARS coronavirus 2 and can be used in the lab to study the new virus. Concerning antivirals, there has been done a lot in the past years, not only on SARS coronavirus 1, but also, for instance, on MERS coronavirus. And one example is the remdesivir that is targeting the polymerase. And it has been now already shown that it also is a promising antiviral candidate for treating of SARS coronavirus 2 patients. So there are a couple of systems that can be used, and I think we don't start from scratch with this new virus.
Thank you, Volker. The virus has a positive strand RNA genome. Can you tell us how long it is?
So coronaviruses are exceptionally long, so the genome is exceptionally long, with about 30 kilobases, and about 2/3 of that is devoted to encode the replicative enzymes, and we call them non-structural proteins, NSPs. And they are encoded in these two large open reading frames that you see depicted as ORF1A and ORF1B. And if you look at the structure on the left side, you see inside the virion the genomic RNA that is wrapped with the nucleocapsid protein. This is one structural protein. And we have in the envelope three other structural proteins: the envelope protein E, membrane protein M, and the spike protein S. And the spike protein is one very important molecule because it binds to the receptor ACE2, but it is also the main determinant for our antibody responses. So most of our antibodies are directed against the spike protein.
Volker, do you want to comment on the furin cleavage site as a key difference between SARS coronavirus 1 and 2?
Yeah, the furin cleavage site, it's still not clear. First of all, why it's there? So it was very surprising. And we still have to do some work to link that to a phenotype. So we actually don't know whether that is important for what people see in the clinics. If that is important for the amazing ability of this virus to transmit so well between humans. But these are questions that should be tackled. And maybe I forgot one thing in terms of experimental systems, which is animal models. And we can rely on mouse models that have been made transgenic to express the human ACE2 receptor, but also other models like ferrets that appear to be a good system for looking at transmission. So maybe the cleavage sites that we see in this new virus can be studied in such an animal model to see if this is related to the ability to transmit better than the SARS-1 virus.
And I think, I agree, Volker. I think I should have said that maybe I wasn't quite clear in my reply to Darius' first question, because you do wonder if the cleavage site may be associated with the amazing ability to replicate so well in the upper respiratory tract compared to SARS coronavirus 1, as you say, that really just replicated deep down in the lung and hence was far less transmissible, although they used the same receptor. But, I mean, we don't really know that yet.
Exactly, and what we also observed now in the lab is that the new coronavirus can grow much better at 33 degrees, while it cannot grow that well at 37 degrees. So there might be also some temperature dependency on, or the reason that this virus is so well replicating in the upper respiratory tract that it loves more the 33 degrees compared to the 37 degree.
Elevated transaminases are frequently observed in patients with COVID-19, but the mechanisms are unknown and may actually be multifactorial, including direct viral effects, effects of the inflammatory or immune response, drug toxicity in some patients, maybe hemodynamic alterations in others, and also other factors. In terms of direct viral effects, Volker, do you think that this virus could infect the liver? Is there any evidence for systemic blood-borne spread of this virus?
So Darius, I don't think there is any solid evidence. So what has been reported is that there is, of course, fulminant replication in the throat, essentially, in the upper respiratory tract. And then in the lung, we don't know exactly whether that is so fulminant, but the virus can very well replicate also in the deeper lung. but from there, it is not clear if there is infectious virus in the bloodstream. So that is maybe not likely. We don't know that from the other common cold coronaviruses. But what has been reported is for quite a long time during the disease and even after the disease that virus can be detected in the stool. And maybe Felix, you can comment on that a bit.
Yeah, it's certain that we can detect it. I would support exactly the same argument you're making for detection of viral RNA in the blood, that while we can readily detect really viral RNA at not even too unimportant concentrations in fecal material, it doesn't seem to be really very infectious, if at all. And I think we can say that at the current state of knowledge, it's certainly not any epidemiologically relevant mode of transmission. So I don't think that clinicians should be wary of acquiring infections when working with fecal material, for example. Whereas, as you say, the replication in the respiratory tract, and notably the upper respiratory tract in the first week of disease, is really predominant and clearly, in terms of human-to-human transmission, by far the most relevant aspect of that virus.
Maybe one little side note on that. So I now get more and more questions from the press and lay people. Can coronaviruses be transmitted via mosquitoes, for instance? And there is so that essentially two answers to that. One is there's not a single coronavirus that infects insects. So they only infect mammals and birds. And the second is exactly what we just discussed. This virus should be, if it's a relevant route of transmission, should be replicating in some systemic way to be transmitted by mosquitoes. So that's absolutely, so we can absolutely exclude this one.
I agree.
Thank you very much. Another question that I would be interested in hearing your thoughts about is the following. We are well aware of the host determinants for vulnerability and severe COVID-19, which include advanced age and certain chronic conditions. But what is known about viral determinants of pathogenicity, and what kind of studies are planned in this respect? Volker, what is your view on that? What is known about viral determinants of pathogenicity?
Yeah, so let's start this question with a very general overview. So we know that the replicase complex, so this is the well-conserved non-structured proteins, there are a lot of proteins that are known to interact with the host cell. And some of them have been already implicated in driving inflammation, but that should be quite similar to other coronaviruses. Then coronaviruses have their so-called accessory genes. So these are not the replicase encoding genes and not the structural genes that end up or the proteins that end up in the virion. So this is a particular set of extra genes, accessory genes. And every coronavirus has its own set. And the more related coronaviruses are, the more similar these accessory genes are. And as I pointed out already, we have the same accessory genes in SARS coronavirus 2 versus the former SARS coronavirus, but they differ a little bit in their sequence, so they may also differ a little bit in the function. And that's of course one question in the field now. Do they have other functions, different functions than SARS coronavirus 1? I think this comparison has to be made now in the near future experimentally. And to answer those questions, we of course can work in cell culture. maybe with more sophisticated culture system like airway culture systems. But ultimately, you have to go into an animal model to answer those questions. And this is yet not clear what will be the best animal model. So far, we see from reports where ferrets have been used that this might be a good model to look at transmission, but maybe not so to look at pathogenesis. And the mouse model, we have to see what this can tell us. This, of course, has then the power of using all the knockout strains that are available to go more into mechanistical studies.
Volker, I think I would comment briefly on that. We are very early still in the pandemic, and we haven't seen a lot of viral variants. Of course, we see mutations very clearly so, but we are yet to see, and I have no doubt they will arise if they are not already circulating. And we will see more viral strains that differ, that may have acquired certain deletions, and we shall see whether those really impact the pathogenesis of the disease. And I mean, we can expect this to happen. One should say maybe at that point that coronaviruses are really different from other RNA viruses. Consider influenza, for example. And Volker, you have mentioned the unusually long genome. It's maybe worth saying that probably because of that long genome, coronaviruses have a proofreading protein that minimizes the error rate, or that it corrects errors done by the viral polymerase during replication. So there is no reason to assume that coronaviruses will evolve at the same speed, for example, as influenza viruses. And I think that that's something that we have to consider, because it will also strongly affect what we can expect of a vaccine and which problems may arise for future vaccination campaigns.
Mm-hmm. Yeah.
Thank you, Felix. Felix, are efforts ongoing at the international level to assemble sequence data on this new coronavirus and to correlate it with clinical outcomes? Can you tell us a little bit about that, please?
Darius, yes, absolutely. It's really great to see there's national and supranational efforts to assemble those databases, and we really need those to get to an epidemiological clue, and also to decide eventually on which strains we should use in the models that are being set up, as Volker just said. And I think this is really vital. We need that information rather sooner than later. And it's not only clinical course of disease and viral genomes that are needed in such a database. We also need to identify, you've mentioned the host determinants of severe disease. So who are those people that do die of pneumonia, of multi-organ failure, when maybe virus is not even present anymore in the patient, yet you see a patient that is dying and you cannot stop it simply because an inflammatory cascade has set on that is not really linked anymore, potentially not linked even anymore to viral replication in that given patient. Of course, I don't want to generalize too much here, but we need that. And what we see is, with the current overwhelming situation where even in very affluent settings, such as Switzerland, Germany, France, we see that our healthcare systems are on the edge of stopping to work, hence all the lockdown and all the difficult situation that people have been facing. We need to identify markers in those databases that help us to stratify patients and to early identify risk patients. And we see that classical markers that in infectious diseases we have been using, procalcitonin, don't seem to stratify patients, or at least not accurately. So yes, of course, we see from the first cohorts, we see differences in cytokine levels, the usual suspects, several interleukins are mentioned in some of those papers, but we are not yet close to a point where we can identify the high-risk patients that we really need to prematurely take to the intensive care unit, that we need to prematurely intubate and ventilate. And I'm really hoping that these strong, large, coordinated efforts will assemble such data, urgently needed data, very quickly.
Thank you.
I think that's an important point, Felix, that let's say the diversity in this virus, in the SARS coronavirus 2, is really, really low. We see it from the beginning that this virus remains almost constant. And therefore, the idea that the virus is changing to cause more severe disease is maybe not true for this virus. It's maybe more the host that is the determinant factor, whether there are other diseases underlying or things like that. And of course, there's one question that can be probably answered in the near future. Is there a difference between the virus that's replicating in the upper respiratory tract versus, I mean, in the same patient versus the virus that is then going to the lower respiratory tract? That would be an interesting study to do.
Now, a last question that interests us, being interested in the liver and being familiar with hepatitis viruses, is whether you think, as expert, that a new hepatotropic virus could emerge similar to SARS coronavirus 2. In this context, Felix, what do we know about the evolutionary biology of hepatitis viruses?
Yeah, what we do know is that it's not so different from the coronaviruses after all. So if you consider the first slide that we looked at, where we saw that coronaviruses generally evolved over millions of years, maybe in a reservoir, bats, for example, not only bats, in many of the coronaviruses infecting humans, we see that in this slide that is shown here, we see on the left the origins of the 5 big hepatitis viruses infecting humans, and in all cases we see animal pictograms. So it's really a similar picture. It's just that the mode of transmission, of course, is different. So if you want something comparable to SARS coronavirus 2 that is causing a pandemic at an absurd speed, then you clearly will only see this in a respiratory virus. But the point I would like to raise here is that the hepatitis viruses were probably all once carried by animals other than humans. So all of the viruses that are known to the hepatologists as major causes of viral hepatitis were once non-human viruses that were acquired at some point that is hard to really reconstruct by humans. And there's one last point maybe I would like to raise here. Although clearly the hepatitis viruses, and especially the blood-borne hepatitis viruses like hepatitis B and C viruses predominantly are far less transmissible compared than SARS coronavirus 2. There's one important point here. Hepatitis B is the prototypic example of an evolutionary strategy of a virus to maintain itself in the human population. And that has implications for us as evolutionary virologists, because consider that if any of these animal hepatitis B viruses infects a human, and if that virus is transmissible via the vertical route, so from the mother to the offspring, then probably, and different from many, many other zoonotic viruses, a single introduction any place in the world would maybe be sufficient if that virus was then subsequently transmissible between humans. So I think we shouldn't discard the possibility that other hepatotropic viruses may emerge, while we should consider that the spread will not be as fast as for the respiratory viruses such as SARS-CoV-2.
Thank you, Felix. Volker, are there any coronaviruses that are hepatotropic already? Are any hepatotropic coronaviruses known today?
Well, actually, there's a very well-known coronavirus since many, many years. We know it. It's mouse hepatitis virus. So you can already see from the name that it's hepatotropic. It's the virus that has been used most for gaining our knowledge on coronavirus biology. It has been used in the mouse model for many decades. And actually, this virus is going systemic. It can infect macrophages. It can infect dendritic cells, so cells that are in the bloodstream and can disseminate the virus in the body. And they have a pronounced tropism for hepatocytes. and cause eventually then hepatitis. And there is actually another example of a coronavirus that is not hepatotrope, but can go systemic, which is the feline coronavirus that is initially replicating in the gut, as many other coronaviruses actually do also replicate in the gut. And upon acquiring mutations, and we don't know yet exactly which ones are really required, this virus can leave the gut. It doesn't replicate very well in the gut anymore, but goes systemic and then cause a lethal disease called feline infectious peritonitis. So there are some examples of coronaviruses that can go systemic, that don't restrict themselves on replicating on epithelial layers, like in the respiratory tract or in the gut, but they can easily, or relatively easy, change their tropism and can go systemic.
Thank you very much. I really enjoyed being able to discuss with you. I think we all learned a lot from you. Thank you, Professor Jan-Felix Drexler. Thank you, Professor Volker Thiel, for taking the time and for sharing your insight with us. There's still a lot to learn. I'm sure we'll see much new knowledge gained over the next months and years, and hopefully new antiviral approaches also based on these new discoveries. Thank you very much.